Adaptive immunity comes into the picture when the innate system is not sufficient against an established infection. In contrast to innate immunity, which is nonspecific, adaptive immunity generates an antigen specificity. The antibodies produced by B cells are so specific that they are able to distinguish two protein molecules that differ in only a single amino acid. The adaptive immune system enables the recognition of an almost infinite diversity of pathogens so that each different pathogen can be targeted specifically. The cells involved in adaptive immunity can also develop an immunologic memory, so that, in the future, if the same pathogen attacks the body, the reaction will be faster, stronger, and more effective in neutralizing and clearing the pathogen. Finally, the immune system can distinguish self from non-self or foreign molecules, and normally responds only to foreign antigens. However, occasionally the system fails to make this distinction and reacts destructively against the host's own molecules, leading to autoimmune diseases. In adaptive immunity, the clearance of a pathogen occurs either by the humoral response, which is B-cell mediated, or cell-mediated response, which is T-cell mediated. Humoral response occurs when a B cell interacts with an antigen. The interaction of B cell with the antigen leads to the proliferation and differentiation of the B cell into a plasma cell that secretes antibodies, and memory B cell, which will take part in future infections by the same pathogen. When an antigen is coated with an antibody, it can be eliminated in several ways. Antibodies can cross-link several antigens, forming clusters that are more readily ingested by phagocytic cells. Pathogens generally bind to host cells through their receptors. These antibodies coat around the pathogen and block these receptors so that they cannot bind to host cells, and therefore the infection is inhibited. This is called antibody neutralization. Cell-mediated response occurs through T cells. Both T helper cells and T cytotoxic cells take part in cell-mediated response. T helper cells are activated when their receptors recognize antigens bound to MHC class II molecules. Activated T helper cells secrete cytokines, that in turn activate B cells, T cytotoxic cells, and macrophages, which eliminate the pathogen by their respective methods of pathogen elimination. The other way of cell-mediated immune response is T cytotoxic cell activation. Cells that are infected by pathogens such as virus-infected cells degrade virus particles inside, and degraded viral antigens along with the class I MHC molecule is expressed on their membrane. T cytotoxic cells get activated when they interact with these complexes. Activated T cytotoxic cells proliferate and differentiate into cytotoxic T lymphocytes. These cytotoxic T lymphocytes exhibit cytotoxic activity, that is, they kill the virus-infected cells and prevent the spread of infection. Gradually, the established infection is reduced, and the pathogen is completely eliminated from the body.